All right, this video is an answer key to the first Hardy-Weinberg practice problem homework sheet. If you're still struggling with this and you had a hard time doing this, don't be alarmed because it's the first worksheet that you've done on this and it's kind of tricky at first. All right, so question A. Rouge snout, also known as Rudolph syndrome, is a dominant disorder found in reindeer. A reindeer with this disorder has a red nose that glows at night. Unlike a rouge-snouted reindeer, a reindeer with normal black nose color can hide better from its predators. A population of 300 reindeer live on the island of Formofurhi. In this population, the allele frequency of the dominant rouge gene is 0.23. Now, normally if we were in class, somebody would ask, is this a real disorder? At which point I would say, of course not. This is a made-up disorder that we created just to have some practice problems. But the allele frequency of the dominant allele is 0.23. So if we have allele frequencies, they go on the outside of the Punnett square. Okay, so the dominant allele frequency goes here by the big N. Now the lowercase n, if it's not dominant, it must be recessive. So basically, in order to get the recessive allele frequency, you say 1 minus 0.23, and if you do that, you get 0.77. The idea with allele frequencies is that they are the percentage of the population that that allele makes up. So if you add these together, 0.23 and 0.77, you should get 1 because it's 100% of the population because the allele is either dominant or it's recessive. Okay, now we put the 0.23 and the 0.77 on the outside of the other part of the Punnett square. Now, if we know the fraction or the percentage of the allele, each allele in a population, we can figure out the genotypic frequencies using the laws of probability that we already practiced in the previous unit. So if we want to know the chances of inheriting two dominant alleles together, it is the product of their individual probabilities. So it would be 0.23 times 0.23, which is also 0.23 squared. And so we get a genotypic frequency of 0.053 or 5.3%. Now, if we want to find the chances of getting the heterozygous genotypic frequency, we multiply 0.23 times 0.77. And then if we want to find the allele frequency of the homozygous recessive, we do 0.77 times 0.77 squared, which is 0.593. So what this means is that 59.3% of the population would be dominant. Now with these, for the heterozygous individuals, we have to add those together. So it would be 0.177 times 2. And somebody usually asks at this point, are they always going to be heterozygous in these Punnett squares? Because we've just previously done the genetics unit and they weren't always. But remember, this isn't a particular couple that are having offspring that we're making predictions about. This is entire populations. So yes, whenever you're doing Hardy-Weinberg Punnett squares, you will always have set it up as though you were doing a heterozygous Punnett square. So the recessive allele frequency is 1 minus 0.23, which was 0.7. The homozygous recessive genotypic frequency would be 0.77 times 0.77 because the individual probability of getting one of the alleles is 0.77. The chance of getting them together is 0.77 times 0.77, which mathematically is the same thing as 0.77 squared. And then it asks what is the heterozygous genotypic frequency, and that would be 0.23 times 0.77 times 2 because there's 2 of those squares in the Punnett square. So the answer there is 0.354. And now it says how many reindeer would be normal. So remember that this is a dominant allele. And so the reindeer that would be normal would be you take the homozygous recessive genotypic frequency and multiply by the total population that they gave you in the beginning. So 0.593 times 300 is 178. So to find, what is 59.3% of a population of 300 reindeer, you just take 0.593 times 300. How many reindeers would be carriers? The answer is none. You can't be a carrier of a dominant trait. So that was a trick question, so be careful. And then the next question says, how many reindeer have the rouge snout trait? And so it would either be 300 minus 178 because we figured out how many were normal and we know the total population. Or another mathematical way to do this would be to take the homozygous dominant genotypic frequency and add it to the heterozygous genotypic frequency, and we get 0.407. If we take 0.053 plus 0.354, we got 0.407 times 300, which gives us 122. Be really careful because the trickiest problem on your quiz is going to be a problem like this. And every year, students will take the dominant allele frequency and just take 0.23 times 300. That's wrong. It doesn't actually work. It seems like it should work. But remember, some individuals are going to be homozygous, dominant, and some of them are going to be heterozygous. And you need to figure out the percentage that would be homozygous, the percentage that would be heterozygous, add them together, and then figure out, multiply that fraction or ratio by the total population to get 122. So this is problem B. Now in this case, if you see a bunch of alleles just typed out for you like this, this is showing you a gene pool. So when you make these counts, you're getting allele frequencies and not genotypic frequencies. So if we count this, we have 2, 4, 6, 8, 10, 12 total alleles. And if you want to find the allele frequency for the albinism gene, which is recessive, you take 1, 2, 3 divided by 12, and you get 0.25. Now, of course, this is a very small representative gene pool, and real gene pools would never be this small, but this is just getting you to see if you know how to do the math. A really common mistake that students make, one of the most common mistakes, is that they take 3 divided by 9. They'll take, there's 3 of the recessive, and then 1, 2, 3, 4, 5, 6, 7, 8, 9 dominant, and they take the recessive number divided by the dominant number, and that is not really how you figure out a percentage. It's got to be 3 divided by the total population. So 3 out of 12 of them are recessive and gives you 0.25. Now, to get the allele frequency for the dominant or normal gene, there are 2 ways you can do this. First of all, you can do it in your head because you know that 1 minus 0.25 is 0.75, or you could take 9 divided by 12. Either way is acceptable as long as you show your work. Now, to calculate the genotypic frequency of albinism, we would take the homozygous recessive and square it. So 0.2, or the homozygous, you would take the recessive allele frequency and square it. So if we were to fill out the Punnett square, we put the allele frequencies on the outside. Allele frequencies always go on the outside, and we're working inwards. So 0.75 times 0.75 is 0.5625. 0.25 times 0.75 is 0.1875. And then 0.25 squared is 0.0625. Students always wonder, where do we round? And it doesn't really matter to me where you round. I always round less and round later because these are estimates, but the more you round early on in different steps of the problem, you end up with your estimate being more and more inaccurate. So never round to more than the nearest 100th, and if you're going to round, do it at the very end. But I usually leave 3 decimal places. If there are 300 people in the population, how many would be carriers? Now this you can be a carrier of because it's a recessive trait. So what we would do is we would take 0.1875 and add that to 0.1875 to get our heterozygous genotypic frequency, which would be 0.375, and then you multiply it by the total population, which they told you was 300. Okay, and then you get 112.5 or 113. Students always ask, should we round up or should we round down? It doesn't really matter to me. I know you can't have half of a person, so people don't like to leave decimals, but these are estimates anyway, so it wouldn't be awful if you left it as a decimal. But if you saw on a test or a quiz the answer 112 or 113, pick the one that's closest to whatever the calculation answer you got when you calculated it. I would never put something like 112 and 113 as an option in the answers. So how many people would be normal? Well, you would take the homozygous dominant genotypic frequency and add it to the heterozygous genotypic frequency. So you add these three boxes together because all of these individuals are normal. So 56% would be normal and or would be homozygous dominant, and 37.5% would be heterozygous, and all of these individuals would be normal. So you add those genotypic frequencies up and you get 0.9375, and then you multiply that by the total population and you get 281. Now, in terms of how many people would be albino, you would just take the homozygous recessive genotypic frequency and multiply it by 300. Or you can take the normal number that we had calculated previously and subtract it from 300, and you get 19 as well. So there are two different ways that you can get this answer. And it doesn't really matter which you use as long as you show your work. All right, this is the trickiest and actually most interesting and fun problem. So we have an island and we have 200 mice that have brown fur, which is recessive, and 500 mice that have the dominant trait, which is black fur color. You need to be very careful because students tend to take 200 and divide by 500. Well, that's not the total population. So the first thing we need to do is figure out the total population of mice that we're dealing with. We have a total of 700 mice. Now on the homework, I asked that question first, just to draw your attention to it, to make sure that you don't make that mistake. But on quizzes, I won't necessarily do that. So if you take 200 divided by 700, you get 0.2857. Now, those 200 mice are diploid. So when we say 200 mice out of a total 700 are homozygous recessive, that's actually a phenotypic frequency and therefore a genotypic frequency. So remember that those 200 mice are not individual single alleles. It's not like you're saying 200 out of 700 alleles. You're saying 200 mice, and each of those mice have 2 alleles. Okay, so we know they are diploid, and diploid organisms, so if we're talking about zygotes, they start inside the Punnett square. So if we know that little b is 0.2857, the trick is, how do we find little b? Now, if you think back to the previous problem, if you wanted to find this number, you would take this number and square it. So now in order to do this, we're going to do the reverse. And so in order to do that, so if b squared is 0.2857, you take the square root of both sides, and the square root of 0.2857 would be 0.5345. Now, once you have one allele frequency, it's very easy to get the other allele frequency and work your way back in. But I will tell you, every year I have students that take 500 divided by 700 and then take the square root and think that that's going to be the dominant allele frequency. The problem is that of those 500 mice, some of them are homozygous dominant, but some of them are heterozygous. So you can't use the dominant phenotype to figure out the dominant genotypic frequency because some of those 500 mice belong here, and some belong here. So you always have to start off with the recessive individuals because you know they're all homozygous recessive. And so once you have this allele frequency, 1 minus this will give you the dominant allele frequency. And then you work your way back in. So if you want to find the homozygous dominant genotypic frequency, it'd be 0.4655 times 0.4655, which is also 0.4655 squared. And then to find the heterozygous genotypic frequencies, you multiply those fractions and add them together. You always want to check to make sure that your two alleles, if you add them together, equal 1. If they don't, what you probably did was take 500 over 700 and take the square root, which you can't do. Also, a lot of times students will take this 200 over 700 and take this number that's right in here that belongs inside the Punnett square and put it out here and then square it when they should be square rooting it. I will tell you that if they give you individuals and they're talking about phenotypes and they're telling you you have whole organisms and phenotypes, you're going to be starting from the inside of your Punnett square, square root, and work outwards. So the allele frequency of the brown allele is 0.5345. The black fur allele was 0.4655. And the heterozygous genotypic frequency was 0.2488 times 2. And so what percent of the population would be homozygous dominant? Well, you just take the homozygous dominant genotypic frequency and multiply by 100, also known as moving the decimal place 2 to the right, and get 21.67%. How many mice would be homozygous dominant? You would take the genotypic allele frequency, or the homozygous dominant genotypic frequency, and multiply by the population of 700. So 0.2167 times 700, because this is our total population and this is our homozygous dominant genotypic frequency. And we get 151.69. I would probably round that to 152. So what percent would be heterozygous? 49.76%. A lot of times I'll get a number that is half of this because students will take the dominant allele frequency and multiply by the recessive allele frequency and forget that there's 2 squares of your Punnett square that you have to account for. So oftentimes students will give me a number for this problem that's half the correct number, and then I know immediately they forgot the 2 part of the 2pq of the equation. Now, how many mice would be heterozygous? We take 0.4976 times 700, which is the total population, and we would get 348.